robust protein and proteoform-level quantification statistical analysis.
Great. Well, first of all, thank you very much, Oliver, and the committee for selecting my abstract. Now, the last question for the last speaker, this is kind of what this talk is all about. So we're talking about protein level and moving on to proteoform level quantification using Bayesian modeling. So we started this project a few years ago because, at least for iTraq, we couldn't find any available methods for assessing differential expression when you're scaling up to large number of samples across multiple iTraq runs without incorporating reference pools. So there's a couple of papers, but the actual algorithms were not available. And of course, MS stats and things like that were for label-free at the time. So we decided to go ahead and develop our own mixed effects model. And we decided that perhaps we could look using Bayesian Markov chain Monte Carlo in order to do the inference, as perhaps we could fit a more complicated model. So first we did this using iTraq and then TMT, and very recently, near the end of this talk, I'm going to show our extensions to label-free. So MCMC is not quick, and this requires quite a lot of computing time. It currently runs overnight on a computing cluster, and I'd say an experiment of 10 or 15 samples is going to take about 8,000 CPU or thread hours of time, which, for instance, on Amazon is going to cost you about £25 to £50 a go. So still in the realms of possibility. So there may be some people in this room, everyone, I guess, knows mass spectrometry, but particular issues with proteomics rather than, say, metabolomics is that we have to digest the proteins down to peptides before analysis, at least with bottom up, in order to reduce the mass and increase the ionization efficiency. And one of the issues here is the digestion process is of cleaving these proteins into peptides using trypsin or whatever is quite, it's a lengthy process, but it's not 100% reproducible. In fact, each one of these cleavage site reactions has a different efficiency, and at the end of the day, each of the peptides that you're going to see in your data set is going to be more or less variable, and some of them may not be good to quantify on. So our Bayesian model takes it's two stages to this at the moment. We've restricted ourselves at the moment to the mixed effects framework rather than full Bayesian possibilities. So in the top left there, this is the first stage is the normalization model, or in order simply to get the scaling factor, the normalization, the bias between samples due to sample loading and other issues. So we actually use a Poisson likelihood model. This was due to the fact that iTraq can typically have very low numbers of counts for the reporters. But as you can see with the exponential there, this is basically a generalized linear model, a Poisson generalized linear model with a log link. So the actual values are still modeled in the log domain, and the normal distributions there in the top right are essentially log normal distributions. So the first fixed effect we have is run channel. That's what we're looking for, the interaction between the run and the channel or label, which is basically our normalization coefficient. Then we have a feature effect, which is just due to the ionization or what's been selected for fragmentation. But then we have a couple of these random effects. The first we call preparation. It's not exactly the sample, because you might have a sample that you prepare multiple times if it's, say, a reference pool. So we make a distinction between a sample and a preparation. So basically here what this third factor here is saying is that we have a normally distributed uncertainty for the preparation, but it's on across preparations, I can say, but this is for each peptide. So if you have, I don't know, 50 peptides in a protein, you're going to have 50 separate variance components in our model that are modeled, and this is then giving you an estimate of how variable each peptide is. At the same in the same way, our error or residual is also log normally distributed, but we fit a separate variance for each. Well, it says spectrum there. It means that was a bit of a typo. It means feature. So each feature has a separate residual variation. So here we have a pictorial representation where there's multiple features for each peptide and multiple peptides for a protein. We're aiming to get the ratios or the normalization coefficients, and we're establishing the most reliable features and the most reliable peptides over this hierarchical model. So we do that separately for each protein. So that gives us a few thousand, maybe, different protein sets of protein ratios, and then it's just a simple median normalization. But actually, it's a Bayesian median. So if we've got 1,000 or 10,000 MCMC samples for each ratio, this gets transported into a distribution for each median as well. So you can immediately see that this is just the same as having a median normalization, but we actually have an uncertainty or a posterior distribution credible interval on each one. And this can help in your quality control. And because we can just do some simple linear algebra so that you get a mean-centered version, so you can really understand actually if your reference channels as well have issues with them. So because this is a mixed effects model, we can actually put in whatever actual experimental design you like, whether it's just case control, one-way ANOVA, two-way, time course, paired samples, etc. And we put this design in our second stage model. So for instance, if we have this 8-run 2-8-plex iTraq experiment here, we can just state that we have 3 separate conditions, and perhaps we're finding we want to know the differential expression between the blue controls and the red STZ rats and the teta, which is a treatment for diabetes. So the second stage model is the same, or is it an extension of the first stage model? The first thing you should note is that the run channel fixed effects that we estimated in our first stage, this is now put into this model as an offset or an exposure for the Poisson generalized linear model, which basically just means that it uses a very strong prior that we generated in our first stage, the output of our first stage. The feature, the preparation and the error terms are the same, and now they're just adding on to that is your experimental design. So this is the most simple experimental design, a condition effect, and then a log normal distribution across your samples, which is effectively the same as a t-test, unless you want to stratify your samples by populations, in which case it's a Welch's t-test. So when you put all that together, at the bottom in green or blue there, we can infer the posterior distribution of the differential expression between the controls and our 2 conditions. And there on the bottom right, you have posterior distributions of the sample variances within our 2 populations, one for controls, one for Alzheimer's disease, for instance. Sorry, that's a typo as well. That should be diabetes. Right, so this now gives you a little view of what's going on in the middle of our algorithm as it's going on. So here's an example based on 7 features across 8 iTraq samples or iTraq channels, and we've got 3 different peptides, 1 red, 1 green, and 1 blue. So you can immediately see that the one off bottom there in blue has a problem with the one of the channels there, the 121, is there's something wrong with it. So during our model fitting process, because we've got an independent, we're estimating an independent variance for that particular spectrum, for that feature, it goes crazy off the scale. The grey is our predictions. We can't predict correctly there, so essentially that feature gets down-weighted virtually to 0 in the resulting protein quantification. If we then look just a view now at the peptide level, you can note that the red and the blue peptides, apart from that one spectrum, have more or less the same pattern, but the green one seems to be taking a different pattern. So what's happening in this case is that essentially the green one is getting down-weighted, the variance is greater, and the actual protein quantification is going to be based mainly off the red and the blue peptides. We can see this at an alternative view now looking at peptides compared to proteins. So the way, of course, it's modeled is that at the top in the grey you have the protein quantifications for each sample, relative protein quantifications, and then now we have 5 peptides, and basically each peptide is being modeled as a pattern away from that inferred protein quantification. So here the middle and the bottom peptide are driving really the protein quantification, the second from bottom and the second from top less so, and the top peptide there doesn't really have much say in the actual protein quantification. So how does this benefit things in practice, the robustness built into our model? So we've been looking at some and developing spike-in experiments. We've developed a spike-in iTraq experiment, but the important thing here that we really wanted to do is that we couldn't actually find a spike-in experiment available that had separately digested samples. They were basically based on digesting your background and your spike in, and then mixing them in already in different quantifications, and then running a bunch of replicates. And we really wanted to show what was more like reality in clinical proteomics, where every sample has to be digested separately. So on the left here we have our first attempt at the iTraq separate digestion spike-in. Now, unfortunately, there was a problem with the sample preparation, so it's actually going to be run again this week, but it does illustrate that on a very hard data set, the benefits of our algorithm. So we compare this in the blue here. We have protein pilot, protein quantifications, and then t-tests on the result of that. And essentially our 1% false discovery proportion, that's our ground truth, which, sorry, I failed to mention that this is E. coli spiked into a rat background. So there's 5,000 identified proteins, about 1,100 that are supposed to be differentially expressed. The protein pilot basically only recovers a few at 1% or even 5% false discovery proportion, whereas our approach recovers about 140 proteins. So that makes it quite useful then if you have quite a dodgy experiment. But while we're waiting for this experiment to be rerun, we started developing, extending our approach to label-free. And to do that, at the moment, we've just used the same philosophy as MS Stats in terms of missing data imputation, because in label-free you have a problem in that your missing data is not balanced between your conditions. So we've added a minimum detection threshold to solve this through a censored Poisson model, and here is the result here. So you can see this is SWOF data, Navarro et al. This is yeast and E. coli spiked into a human background, and at 1% false discovery proportion, we're getting slightly less than double the number of proteins being called differentially expressed. So coming close to the end of the talk, what we're doing now is looking at our FDR calculation. At the moment, we just calculate a local FDRs based on the posterior distribution, but it's not very well calibrated, and we're developing an alternative Bayesian model comparison with Bayes factors approach, which looks at first instance to be working quite well. One of the nice things about getting individual peptide estimations of peptide variance is that we'll be able to create a large atlas of peptides from this collaboration we have with the University of Manchester. So this could help quite a bit when you're looking to develop mass spectrometry assays. And then finally, we've completely, of course, ignored shared peptides at the moment, but they're really obviously prevalent if you want to look into modeling proteoforms, genes, particularly with gene isoforms and proteolysis products. Our version 1 model works very nicely. For instance, for amyloid precursor protein, there's this one or two particular peptides here that are crazy, and we reject them as expected. But it turns out, for instance, that this is a subprotein, amyloid beta, that's been associated with Alzheimer's disease, and it's showing a very different pattern. So right now, we've gone on to develop a mixture model in order to extend this to true proteoform modeling. And with that, I'm going to end this talk. The team is myself and Ranjit, who's done the cluster computing, and Alex, my PhD student, who's doing the proteoform modeling. Thank you very much for your attention. The actual thing is the scaling issue is really on the number of peptides and features per protein. So for instance, usually the first or the most prevalent proteins in your experiment, the first one or two, may have several hundred peptides, several thousand features. And this can take up 10 to 15% of our overall computing time.
So it's a large subnetwork set, basically.
Yes. And of course, if all you want is proteotypic differential expression, you could argue that you probably don't need to model absolutely everything. So there's plenty of ways to reduce that computation. But also, I don't think the aim of this was to see what was possible with Bayesian modeling, but there's plenty of opportunities with, say, variational Bayes. We have a collaborator who works on sequential Monte Carlo that could speed this up considerably.